Allison Burrell. Welcome to the Radiation Research Society Vodcast. I'm here with Jodi Belz. And Jodi, so I hear you're doing a research project on slow release of docetaxel in, what is it, the brachytherapy spacers. Brachytherapy spacers to treat prostate cancer, right? To radiosensitize the prostate cancer cells prior to radiation. Mm-hmm. So would you tell me a little bit more about brachytherapy? Because that's really interesting what they do with these spacers.
Yeah, so currently in brachytherapy, clinicians will inject dozens of radioactive seeds directly into the prostate itself. And these radioactive seeds are spaced out with little plastic spacers. So these spacers are really just to prevent the radiation from overlapping with one another and for the spatial guidance when the clinician's injecting, so there's little tick marks on the needle. Mm-hmm. So what we want to do is replace those plastic seeds with a degradable seed that will release docetaxel directly into the prostate and then have the docetaxel release slowly while the radiation is acting slowly over the course of 60 or 80 days, whatever radioactive seed you have. And then the docetaxel will radiosensitize the prostate during that whole brachytherapy course of radiation therapy.
So in brachytherapy, these radioactive seeds, do they degrade as well as the spacers?
No, so these seeds are for low-dose radiation. They're typically permanent implants that will just have a half-life of, so for I-125 seeds, that's the most common brachytherapy seed, it has a half-life of about 69 days. Okay. So what we're aiming to do is have these degradable seeds, or degradable spacers, so that the total release of docetaxel will be during that time. So we have a nice linear release to radiosensitize. So right now, clinicians will usually give docetaxel IV treatment for metastatic prostate cancer. So what we're aiming to do is prevent that need for the IV treatment and deliver it locally there, because one of the great things about docetaxel is it's not only a chemotherapeutic, but it's a radiosensitizer. So the way that it works is it freezes the cell in the G2M phase, which is the most radiosensitive phase. Mm-hmm. So it inherently is a radiosensitizer. So we expect it to treat the cells and also pre-sensitize them during the radiation.
So are you already doing this in clinical trials?
No, so right now we're still in preclinical studies. What we first did with our research is we wanted to show that delivering it locally was a feasible way of delivering docetaxel. Right now it's delivered in the IV formulation Taxotere, and it's been shown to be pretty effective at high doses, but there's also a lot of systemic side effects. So we did a one-time dose equivalently IV versus the IT directly in the tumor. And we showed that at the same dose one time, we were able to completely cure over half of our mice with the intratumoral injection, whereas our IV mice had a lot of toxicities, they had a lot of weight loss, and initially they responded to the treatment, but pharmacokinetics show that the drug only stays in the body for no more than a few days, so all the mice ended up with tumor recurrence and none of them were treated. So we really showed that delivering it locally was a feasible way, and then we moved on to combining it with the radiation to see if at too low doses we could have a synergistic effect. And that's what I'm currently working on now. We've shown that at half of the dose that we use to treat the mice, when you combine that with fractionated dosing of radiation, we were able to, it's still ongoing, but we were able to cure about 50% of our mice so far. And this is 150 days out.
That's amazing. And I forgot to ask you, what university are you associated with?
Oh, so I'm currently at Northeastern University in Boston. I work with Dr. Sridhar's group, and we work closely with Dana Farber, the radiation oncologist over there, so that we can really get the clinical translation aspect of this, because you don't just want to do research and not have it go anywhere. Right. So we've started off using nanoparticles, but we've ended up moving closely just towards FDA-approved materials so that we can make it easy to translate into the clinic for future.
Now, is this something that's been done before, using these degradable intratumor brachytherapy spacers?
So intratumoral spacers or intratumoral implants have been used before. This isn't a new idea, but this application of it is the first of its kind. No one's really ever used an implant that will degrade slowly in order to radiosensitize. So our lab has shown that you can incorporate a number of different agents or molecules or nanoparticles that can sensitize the prostate or pancreatic cancer or breast cancer when you combine it with radiation. So what we do is we pre-treat these tumors, we let the spacer start to degrade, the drug or nanoparticle, whatever it is, will slowly release into the tumor, and then you can hit it with radiation. And we've shown that we can have really great synergistic effects with that.
So normally these spacers are still used even when you're just doing regular brachytherapy. Are the spacers just like the radioactive seeds permanent implants, or do they normally degrade? Or is this is also a new technique, having it degrade?
Yeah, so I think what's currently used are non-degradable spacers. It's biocompatible, but I think recently they've started to move towards degradable spacers. There's a lot of degradable polymers out there, and clinicians are really trying to stay away from any sort of permanent implant. Exactly. So that's why if we slowly have it degrade, we're using PLGA, which is an FDA-approved material. It's commonly used as a biodegradable implant or nanoparticle. So it's really well characterized.
Yeah, if you can prevent any possible side effects or long-term effects, that would be great.
Yeah, so this completely resorbs into the body and will degrade through natural metabolic processes.
So what kind of techniques are you using to evaluate the efficacy of this procedure? Are you just looking at the size of the tumor? Are you using any markers or DNA damage markers to evaluate how well it's radiosensitizing the cells? Sorry, that was a lot of questions. But
so the study is happening now, so we haven't gotten that far yet because most of our mice are still alive. But when we do start to sacrifice them, we'll do some tunnel assays to measure the apoptosis in the tumor and look at the caspase. And then we're going to look at some gamma H2AX and see the DMA damage that is experienced. But we've now irradiated the cells 150 days ago. So it's really trying to compare the markers that are present in the mice that were irradiated that long ago and versus the ones that were pre-sensitized with the treatment.
Right, because gamma H2AX, you're not going to see
we're not gonna
that
really
far
see
out.
Exactly. So in our next study, we're going to do a toxicity study, and we'll be measuring those markers immediately after treatment, along with any sort of blood chemistry and toxicities, so we can measure whether or not our implant is causing any adverse effects. And we really want to show that when you do the treatment locally, it will prevent the systemic toxicities that you're seeing when you do IV docetaxel with radiation, because even though you try and do targeted radiation, there's still a lot of healthy organs down there that we're really trying to bypass.
Exactly. And so as far as typical treatment for prostate cancer, does this docetaxel and radiation combination therapy, is this a pretty common way to treat it? Does it only work with certain types of prostate cancer?
Yeah, so prostate cancer is really slow growing, so there's a few ways to decide what treatment route to go. So if it's very slow growing, they'll do a watch and wait scenario, and then they kind of assess it every year to see what the next treatment should be. But for aggressive prostate cancers that are usually the ones that are deadly, a prostatectomy would be used where you remove the whole prostate in combination with radiation. And then as it starts to metastasize, they'll add in this chemo agent, docetaxel. But here what we're trying to do is prevent the spread of the disease before it happens. So, and really for patients that have no other treatment options, where maybe you've already received a prostatectomy and your cancer has recurred, and the prognosis does not look so great. So you really want to tackle it from all angles, because the cancer is mutating and now adapting to these first lines of treatment that you've already seen, and now it can become unresponsive. So a lot of the first lines of treatment, while they're great and we have early detection now, they still are not giving us the quality of life that we want to see in patients down the line, and that's where a lot of these treatments are failing.
Right. So do you ever take into account like pharmacogenomics, like looking at, precision medicine, looking at each patient's tumor individually, and what its genetics are, and then using that, knowing, this treatment works better with this mutation set?
Yeah, so that's something that we would definitely get into when moving into clinical trials. You need to carefully design your trials so that they succeed, and that's where we'd want to look at patients that have the right mutations that we know would have a better shot at responding to this treatment. But docetaxel, it is a microtubule stabilizer, so it's pretty genetic independent. There are cell lines that are resistant to it, but a lot of them will respond initially. And we see that with, if they just came out with a clinical trial that showed when you start to feel bone pain and then you administer docetaxel before it even shows up on the scans that you've metastasized, they've shown that you've had a lot better median survival times and that your treatment ended up being far superior than before you didn't administer it. So I think this is really something that you want to listen to the patient and look at the patient's genetic profile and see if they're really a right candidate for this sort of option, as with any sort of treatment. Now everyone's moving into personalized medicine, which I think that's really the future.
Exactly. And so you had said, docetaxel was the main chemo that you were using in these spacers, but you also said you could add other types of drugs. So once you do this proof of concept, is it pretty easy to switch out different types of medication within those spacers to accommodate people that might not respond or aren't responding to docetaxel?
Yeah, so the way that the FDA would approve the drug, it would have to be classified under a new drug delivery agent. Okay. So we'd still have to go through clinical trials, but as a proof of concept in our lab, we've shown that you can change out docetaxel. We've done BMN or talazoparib, a PARP inhibitor, and we've shown that that can cause a lot of DNA damage that we weren't seeing when it was given orally. Or we added in gold nanoparticles to radiosensitize pancreatic cancer. So you'd still have to create them all as their own individual, but changing out the molecule changes the degradation kinetics and how it releases, and everything is different hydrophobicity and if it's going to degrade at the right speed. So you'd still have to tailor the spacer to the exact treatment that you would want.
So you mentioned pancreatic, so your main project is working on prostate cancer, but this brachytherapy and spacers can be used in other types of cancers?
Yeah, so we're looking more at using the spacer as a sole monotherapy that you can inject into maybe non-resectable tumors and then add outside radiation or external beam radiation to come at it from two different ways, doing the chemo and the radiation, or sensitizing it and then adding the radiation. But brachytherapy, as we mentioned before, people don't really like the permanent seeds, and it's used in very limited situations. Now they have high-dose radiation brachytherapy where you can actually go in with a probe, irradiate the spot that you're looking for, and remove it. So these permanent seeds are very limited in their application, and it's great where we can use these spacers, but this is really just a platform that we can do a lot of different things with it.
Off the top of your head, other than prostate and pancreatic, what are other types of cancers that this might be used on?
So right
Solid
now,
tumors,
so we've been doing a lot of work with talazoparib for breast cancer in these spacers, because right now PARP inhibitors are given orally and they have really bad bioavailability and pharmacokinetics, so the patient has to take tons of pills every single day, and it ends up, for such potent PARP inhibitors, it ends up giving bad side effects and treating off-site toxicities. So we gave it directly into a metastatic breast cancer model in mice, and we've shown that the primary tumor ended up responding a lot better with the local treatment than with the oral gavage. But again, you're really only treating the primary tumor that you put the spacer into, so it's very limited that way.
Right.
So you still have to know your limitations. So in prostate cancer, if you do have micrometastases, you can still deliver docetaxel intravenously, but you can also radiosensitize the prostate using this thing. So it would really just be an added-on treatment.
Right. Do you see in the future possibly doing a combination of two different drugs within the spacers?
Yeah, we've actually just been talking about that in our lab, trying to figure out what combination we might want to do, what dosing you'd want to do. And the only challenges that I see there are for radiosensitizing, you want to have the drug there before the radiation. And in some drug combinations, you want one available before the other or vice versa. So it really depends on what the best treatment delivery schedule is, and then we can design it that way. But we've definitely been thinking about doing that.
It's very interesting.
Yeah, because then it's just a one-time delivery. You don't have to go in for a few weeks or months and then maybe go back and get another injection, rather than the every 3 weeks going in for hours of chemo. So it's a really cool option, I think.
Being able to have these spacers release the drugs at different times within the same spacer, or would it be two different spacers, one that degrades quicker than the other?
Yeah, that would be the two options that we're deciding between.
This is a bioengineering
exactly.
problem too.
I think it would be more feasible to deliver them separately, because then if you need to tailor the dose to the person, then you can change them out. Right now we're giving doses by the length of the spacer, so we have zero order kinetics of our release, so no matter how long you cut your spacer, the same amount of drug is being released every single day, which is really important for scheduling. And when you're trying to treat a mouse and then move into a human, you want your release kinetics to stay the same. Right. So what we would end up doing is, right now we have 2 millimeters of spacer. Moving into human, we would have 5 to 8 millimeters, and that would just be a higher dose that the patient would receive every day, and that would translate. So if we needed to combine multiple drugs into the same implant and then the dosing's not correct, we'd have to start at square one.
This is a technical question, but is the dose associated with the size of the tumor or the BMI of the patient? Or
the BMI of the patient. Okay. So right now for docetaxel, they're giving it 75 mg per meter squared of the patient. And then we're doing half of the maximum tolerated dose in a mouse. So it's really dependent on the size of whatever you're treating. But that, again, we can scale easily with our spacer.
Yeah. So what's interesting is that I can't remember if you said it was docetaxel or if it was a different medication that giving it orally didn't have any effect on DNA damage or increasing the radiosensitivity of the cancer cells, whereas when you gave it intratumorally, you did see that DNA damage. So are you going to, in the future, possibly look into different types of drugs that, I mean, you wouldn't even think to use because they don't have any effect orally or intravenously?
Yeah, I think that's where now nanomedicine has really brought back a lot of drug options that have failed in the past because delivery was not optimized or the toxicities were too high. So now people are trying to put the drugs in a nanoparticle to protect the body from the particle, or protect the body from the drug and the drug from the particle. And here it would be the same thing, where you could deliver maybe something that isn't as potent or has a lot of bioavailability issues or pharmacokinetics or toxicities, where now you can directly put it into the tumor and you don't have to worry about all of those things. Right. So I think this leaves a lot more room for options. We'd still have to investigate drugs that aren't necessarily in the clinic yet, but maybe in clinical trials. But that's why researchers work closely with clinicians to see what they think is best and get the actual impact of what they're doing.
Is your PI an MD or a PhD?
He's a PhD in physics, but he's now, in the last decade, moved into the nanomedicine realm, and he started a lot of teaching grants at our university, but he's been very instrumental in setting up these collaborations, because we're right in Boston. So he's had a lot of collaborations in the city, at the hospitals, making sure that all of the students get in the door with the clinicians so that we can really understand what is important and what we're doing.
All right. Well, I guess, is there anything else you'd like to talk about your project?
No, I think it's really something that can be really translatable, and I think that's what's important. While like basic research is really crucial in understanding how cancer progresses and how patients might respond, we really need drugs that can get to patients now. and this was really designed so that a patient can use it.
Okay. Yeah, I'm very excited about this project, and I love its translational nature. Hopefully we'll talk to you again in a year or so and see where it's going.
Yeah, hopefully it's gone a lot further in a year, and next we're going to move into large animal models and test out the real pharmacokinetics and toxicities associated with it so that we can move into clinical trials.
I look forward to hearing more. Thank you, Jodi. Thanks for joining us.
Thanks for having me.